I nicely follow on from Violetta and Catherine because I'm investigating infection risks in antelope reintroductions. And I'd like to start by explaining why infectious diseases are important in reintroduction programs. And so land sharing approaches to conservation mean that humans and their livestock share spaces with wildlife. And it's clear that elements of land sharing will be necessary to create sustainable populations of Sahelo-Saharan antelope. And this clearly links in with the conference theme on human-wildlife interactions. And for infectious diseases, the wildlife-livestock interface is particularly important because antelopes share many diseases with livestock. And this is why it's really important that we follow the One Health or eco-health concept. And I was really pleased to see this mentioned in Sahara Conservation's strategic plan. And this aims to achieve sustainable human and animal health and well-being through healthier ecosystems. And it requires us to integrate our understanding, investigation, and management of health between species and across ecosystems. So, thinking about reintroductions, they can alter the species balance in shared landscapes, and this can apply to the pathogens as well as the hosts, so as well as the big animals we think about. And this can potentially lead to changed infection risks. And this diagram illustrates that these shifts can lead to disease outbreaks and pathogen spillover, which can also impact on human food security, especially in the Sahelo-Sahara region, where many of the people are heavily dependent on their livestock for their livelihoods. And whilst we often hear about emerging infections coming from wildlife and spreading to humans, it's really important to think about population density because that's really crucial in infectious disease dynamics. So often wildlife populations aren't big enough to sustain pathogens on their own, whereas infected livestock reservoirs can mean that wildlife species that otherwise wouldn't maintain the pathogen can be affected by these diseases, and that can lead to biodiversity loss. And it's therefore really important that we use these One Health approaches to try and ensure that reintroduced populations and the wider ecosystems are sustainable, whilst minimizing the risks to livestock and humans. And it's really important that we think about infection risks, both from wildlife to livestock and livestock to wildlife, and how this all impacts on humans. So the objectives of my research are to evaluate the health status of antelope. My PhD primarily focuses on scimitar-horned Oryx, and particularly the translocation and reintroduction programs in Tunisia and Chad. But sometimes I've collected samples from other wild ungulates where they're available. And the plan is to compare the health status of the antelope with local livestock, to identify the infections which pose the greatest risks, and then to also collect and store samples so that they're available for future reference. So my talk today will focus on fieldwork conducted in the Wadi Rime Wadi Ashim Reserve in Chad over the past 6 months, as this is the only site with wild free roaming oryx directly in contact with livestock. And you can see in these photos the direct contact that can occur between the oryx with sheep in the bottom right and camels in the top right. And then also that these animals will use the same trees for shade. And often these trees have ticks that then accumulate there looking for blood meals. And this is a great way to spread infections between different species. Where there are livestock, there are also humans. And particularly at resources like wells, the population densities can become larger, which, like I said, is really important in infection dynamics. So in terms of the fieldwork, it was part of a program to anaesthetize animals to place GPS collars. And we collected health surveillance samples with the help of the team from Environment Agency Abu Dhabi and the field monitoring team in Chad, who located and identified target animals. And here you see the rest of the herd wondering why their friend has just fallen over when it's been darted. So once the animals were anaesthetized and restrained, quite a big team came in to place the collars and collect samples. And really important was the collaboration with the local vet teams at the research institute, the government research institute in N'Djamena. And you can see some of the colleagues on the picture there. And in this picture, I'm collecting swabs at the front end, while my colleague Cherry Dungus is collecting a fecal sample at the other end. We also collected some samples for genetic surveillance, including fecal samples from the crates for a microbiome project, and some environmental fecal samples and buccal swabs for ongoing population genetic monitoring, which fits in with some talks by Emily and Cara yesterday. So some of the technical challenges many of you will be familiar with. This is the fridge freezer in the back of the truck, being powered by a solar panel to try and maintain cold chain for our samples. And these are the lab facilities we were using. So in the top right picture, the building or the container on the left is the lab. And these are pictures from the inside of me processing samples and extracting DNA in the middle of the night in the Sahel. That was quite cool. But fortunately, through our collaborations with the laboratories in town, on the second trip, we were able to transfer those processes to the laboratory there and use their much better facilities. And this is me working with Didi, one of their PhD students, to heat treat and extract DNA to make the samples safe to bring back to the UK, and also to, really importantly, store a set of samples in Chad, both for kind of future reference, and then hopefully in time, more and more of the processes will take place in country. And one really great moment on the trip was when the Livestock Research Institute thanked us for creating the project, because it was helping them develop links with the wildlife department of the government, because they really see the importance of that going forward with the requirement for One Health approaches. So to date, we've collected 27 samples from 27 animals for health surveillance, and a further 65 for the genetic surveillance in Chad. And that's been augmented by a sample set of 32 animals that Marie Petretto collected in Tunisia. And it's really great that of those samples, you'll see quite a few were born in the wild, 12 born in Chad with 5 genuinely wild borns. The others were born in the pens. In terms of methods, they'll be both looked at current infections through next generation sequencing, as well as traditional serology approaches, to look at a broad range of infections. And then future work is being carried out. So a cross-sectional survey is planned in livestock in September in Chad, and there might be opportunities for livestock sampling in Tunisia as well. And it would also be great to collect samples along the full period of reintroduction, so post-arrival, but before release, as well as after reintroduction. And I can see Pierre's lurking, so I'll just quickly wrap up. But hopefully this will give us a better understanding of the health status of these animals and enable us to identify One Health solutions to manage both the animals going forward and the wider ecosystem to try and make these projects sustainable. And I have a huge number of people to thank, and I can't possibly mention them all, but thank you so much to my funders on the right, particularly Marwell Wildlife, for all their help and support. Catherine Merters at Smithsonian, who shared the vision for the project and helped make it possible, and obviously Sahara Conservation for all their help, as well as all the in-country agencies who've been amazing in terms of collaboration, logistics, and giving me permits. So thank you so much to everyone for their help.